So let us look at what? The structure of bacteria. So if they are talking of the structure of bacteria, So assume that this is a what? A bacteria structure. So that what is a bacteria what? Structure. Like this. This first layer you see here, let's call it what? Cell membrane. Are you getting it? And the next one, we have what? Another layer here that surround the what? The cell membrane. This layer that surround the cell membrane, I would like to use this red marker to segment it. The segmented one here that I'm trying to what? You can see it like this. To segment. This is what we call that segmented one. Yeah. The have there is what we call peptidoglycan. Look at the peptidoglycan. Let me help you to break this to syntax. If we have the word peptide in Greek, means protein, what do I call it? But look at the word glycan. Glycan means what? Glucose. So the peptidoglycan cell what? Cell wall simply means what? Is protein glucose cell wall. And after that, we have another word here. This one is what we call outer cell membrane. So this one is inner cell membrane. The second layer here, we call it what? Outer cell membrane. And the next thing is, in the outer cell membrane, there is a particular what? Surface, which is look like ear-like structure, and we surround the entire surface of the bacteria, and this ear-like structure, short and hair-like structure, is what we call pili. So this structure here, we call it what? Pili. Pili is a singular. Is the one that I said is used for what? For conjugation. Kind of asexual reproduction is used for the attachments, and also transmits what? Genetic material from one bacteria to another. And the next one here is long than bacteria himself. This man is longer than what? Bacteria in what? Himself. Can you see it? So this is what we call what? Flagellum. So the flagellum is the locomotory organ in what? In bacteria. It's used for what? For locomotion. In what? In bacteria. And the next thing we have in bacteria is this. At the center of bacteria, there is a genetic material there. These are the genetic what? Material we have in what? In bacteria, which is what? A nucleus. And we cannot call it a true nucleus due to the what? There is no nuclear membrane here. So this represents what? Nucleus, which contain genetic what? Genetic material in bacteria. The next thing is, in this bacteria, there is a what? The inner folds of what? Of cell membrane. It will fold like a finger. It will fold like a what? A finger in the what? In the cell. Like this. It will fold like a what? A finger in the what? In the cell. And this thing that is folded like a finger inside the cell of bacteria is what we call mesosome. Mesosome. This mesosome is the one that is used for the what? Respiration. In what? In bacteria. And I said it's equivalent to the what? What we call mitochondria in eukaryotic cell. And again, the bacteria, I said, they lack what? Double membrane organelle. But the only organelle we have in bacteria is ribosome. It have what? Ribosome inside the what? By what? Bacteria. There are many. So this is what? What? Ribosome. Ribosome is used for the protein synthesis. And it also produces what? DNA and what? And RNA. Cell wall, it gives shape to what? To bacteria. And it ought to protect the internal structure of what? Of bacteria. And it's also used for ground staining in the what? In the bacteria. Yes, ribosome. And the next one is what? The entire surface of bacteria, the body, here is what we call it what? Cytoplasm. What will I call it? Cytoplasm. Cytoplasm is used for metabolic reaction. So this is what the structure of what? Bacteria. But let me add to this flagellum. This flagellum of bacteria is comprised of three things. Three regions. Yes. It's comprised of what? Of three what? Region. The first one, we have filament. The second one, we have hook. The third one, we have basal body. Please, what is the filament? The filament, this is flagellum, abi? At this side of flagellum, here, this side is what we call hook. So the upper part here is what we call what? What? Filament. And here, inside here, we have what we call what? Basal what? Basal body. So it's the basal body that's attached flagellum to the what? To the cytoplasm of what? Of bacteria. And this hook is the one that is what? Attached filament to the what? To the basal body. It's joined basal body and what? Filament together. And this flagellum, I said is what? Is used for the what? For the locomotion in what? In bacteria. Abi? And the flagellum used for what? Locomotion in bacteria, and is contained. There is a what? A particular protein in this flagellum which is control the flapping movement of what? Of flagellum. We call it what? Flagellin. The protein particle that is present in flagellum, which is making to what? To control the what? The locomotion movement, is called flagellin. The protein part of flagellum is called flagellin. And this upper part here, we have here, this segment here. Our part here, everything we have here like this is what we call what? Filament. So that is all about what? The structure of what? Of bacteria. And the next thing is what? Let's look at what? The classification of bacteria based on what? The types, or based on what? On flagellum. No, it's not all bacteria that have flagellum. I said it earlier now. So that bacteria that have flagellum are called trichos, but from bacteria without flagellum are called atrichous bacteria. So let me pick up the board and move to the what? The classification of what? Of bacteria with, or let me use this place for it. So let me use this site for it now. Classification of bacteria. Classification of bacteria based on what? Flagellum. Good. See, we have some bacteria that is have flagellum. We have some bacteria that they don't have what? Flagellum. Any bacteria that have flagellum are motile. Any bacteria without flagellum are non-motile. So the bacteria with flagellum, we call them trichos. Trichos bacteria. Trichos bacteria is a bacteria with flagellum. But we have what? Atrichos bacteria is a bacteria without flagellum. Please. Any bacteria that have flagellum are called trichos. Any bacteria without flagellum are called atrichos. And a bacteria with flagellum cannot be classified into these following parts. Look at it here. We have bacteria shape in this form, but a bacteria that is have what? One flagellum at one end. That bacteria are called monotrichos. What do I call it? Mono what? Trichos. A bacteria with one what? Flagellum at one end. But if you have what? A bacteria that is have, we have bacteria like this, and they have what? Two flagellum at opposite what? End. One here and the other one here. Any bacteria that have what? Two flagellum at two ends, opposite end, we call it what? Amphitrichos. Amphi means two, amphibian. Amphitriotic Trichus. Amphitriotic. And we have some bacteria that they have what? Flagellum. We have bacteria and they have cluster flagellum at one end. If flagellum are what? There are two or more flagellum at one end of what? Of bacteria. We call it what? Cephalotrichus. We call it what? Cephalotrichus. A bacteria with two or more flagella at one end are called cephalotrichus. But we have a bacteria that have what? We have a bacteria like this and have two or more flagellum at the two ends. What are we call it? We call it what? Lophotrichus. Lovo what? Trichus. And we have what? Some bacteria that they have flagellum in their entire body surface. Like the one we have in what? Like a pili. You know, pili, I said, is a hair-like structure. They are short, but flagellum, they are about most times flagellum are longer than what? The bacteria itself. So a bacteria that is surrounded by what? By flagellum in entire what? Body, we call it what? Peritrichus. So these are the what? The trichus classification of what? Of bacteria. Any bacteria that have one flagellum, monotrichus, two at two end, amphi, cluster at one end, cephalo, cluster at the two opposite end, lophotrichus, all the entire surface we call peritrichus. And the last aspect of classification of what? Of bacteria based on what? On a ground stain. Yes, classification of bacteria based on what? On ground what? Ground stain. So classification of bacteria based on ground stain. What is a ground stain? Ground stain is the process of adding dye into the cell wall of bacteria. The process of adding dye into the cell wall of bacteria is called ground stain. So in terms of what? Of this ground stain, bacteria can be grouped into two parts. We have what we call what? Gram positive stain, and we have gram negative stain. Gram positive stain bacteria. Gram positive stain by what? Bacteria. They have they have a thick, abi, they have a thick peptidoglycan cell wall, which retain mm, retain retain the crystal violet uh, retain crystal violent stain. Crystal violent what? Stain due to presence of teichoic acid. Yes, a teichoic acid. Hmm. Uh, and they always appeared purple under microscope. And they what? They appear purple under microscope. Look at what? This polygram stain. The polygram stain bacteria, they have thick what? Cell wall. Assume this is the cell wall of peptide what? The peptidoglycan. Can you see it? Assume this is their cell wall. The cell wall for positive stain bacteria. This is the cell wall. The cell wall we have here is thick. They have what? A thick cell what? Cell wall, which is what? Peptide what? Pseudoglide what? Glycan. So the presence of this thick peptidoglycan cell wall is to make what? A positive grain bacteria, gram bacteria, to retain the what? The crystal dye on it and appeared purple. But in case of what? Of other bacteria, this sort of, look at the structure of this one. For that one, we have something like this structure. We have something like this structure. We have what? The cell wall will be in this form. Look at the cell wall. This is how the what? The cell wall of negative stain bacteria be. Look at this. See one is thick. The cell wall in here will be what? Will be thin. They have thin cell what? Cell wall due to the absence of what? Teichoic what? Acid. This teichoic what? Acid is the one that is control the what? The thickness of the what? Of the cell wall and dye retaining. So this one lack work. Thin cell wall without teichoic what? Acid. And here, this cell wall of this negative bacteria, it will not have a thick layer here. This thick layer we have here is comprised of what? Of lipid by what? By layer. And this layer here is a lipopolysaccharide layer. Can you see it now? So for the negative stain bacteria, they have a thin peptidoglycan cell wall which cannot retain crystal violet stain due to the absence of teichoic acid, and it appears pink under microscope. A sample of this of negative stain bacteria we have E. coli, but a sample of positive stain bacteria we have staphylococcus and streptococcus. The example of what? Of this what? Of this by what? Bacteria. Am I communicating? Yes, but look at what? The negative stain bacteria, most of the negative stain bacteria, they are the most deadly disease caused bacteria because they have what? High resistance to what? To antibiotics. What makes them resistant to antibiotics? Due to the what? To this what? Extra layer they have. Look at this one that give us what? Positive stain. Yeah. They have what? Just thick peptidoglycan, no extra wall. But in negative strain bacteria, what? Bacteria, they have what? A thin cell wall, this is the thin cell wall in the middle here. They will now have extra layer which comprise of lipid and polysaccharide component. So the presence of what of this lipid layer in the negative cell bacteria, it will make them to be what? To have high resistance to the what? To disease. And a lot of that, they cause dangerous or deadly disease to what? To human. An example of it we have the what? E. coli. So the negative strain bacteria is the inverse of what? Of this. They instead of thick, tiny peptidoglycan cell wall, which cannot retain crystal violet stain due to the present absence of teichoic acid, and they appear pink on the microscope. So the next is what? Shape of bacteria and the arrangement of bacteria. After that, we go to the what? The bacteria growth curve.